Dr. Rosita and friends. Today we are from Group 9 and we are going to present Signals Cascade Targeted Stem Cell Therapy for Lung Cancer. Okay, what is lung cancer? Lung cancer is a cancer that starts in the lungs. When a person that has lung cancer, they have abnormal cells that cluster together to form a tumor. And also, this lung cancer, known as bronchogenic carcinomas, that arise from bronchi in lungs. There are two types of lung cancer, which are small cell lung and also non-small cell lung cancer. This small cell lung cancer is a disease in which the malignant or cancer cells form in the tissue of the lung, and this type of cancer is more aggressive, and the cancer tends to grow quickly and travel to other parts of the body easily, and the survival rate tends to be low. There are a few risk factors that causes the lung cancer, which are smoking, asbestos, toxic chemical, radiation exposure, second-hand smoke, and also genetic. There are four stages in the lung cancer. The first one is the cancer may be present in the underlying lung tissues, but the lymph nodes remain unaffected. The second stage, the cancer may have spread to nearby lymph nodes or into the chest wall. The third stage, the cancer is continuing to spread from the lungs to the lymph nodes or to the nearby structure and organs. And the fourth stage is the cancer has metastasized or spread beyond the lungs into the other areas of the body.
The Wnt signaling pathway plays an important role in the regulation of various cellular processes, including cell proliferation, cell migration, and cell survival. Specifically, in non-small cell lung cancer, it is found that the Wnt protein expression also associated with high tumor proliferation and poor prognosis. This pathway is activated when the Wnt ligands bind to the Frizzle receptor that leads to the stabilization of beta-catenin and its nuclear accumulation. During this process, the APC complex is permanently disabled and promote nuclear accumulation of beta-catenin. This Wnt signaling can be classified into two, which are the canonical Wnt pathway and non-canonical Wnt pathway. During the absence of WNT in canonical pathway, a beta-catenin destruction complex is assembled which consists of axin, APC, GSK3-beta, whereby beta-catenin is then phosphorylated and proteolytically degraded. If WNT is available to bind to the Frizzle receptors, then a receptor complex between WNT, FZD, LRP, DVL, and axin is formed. Then DVL becomes phosphorylated and eventually inhibits GSK3-beta, consequently stops the proteolytic destruction of beta-catenin. The beta-catenin subsequently accumulates in the cytoplasm and migrates to the nucleus to form a complex with TCF and LEF transcription factors. Then, the target oncogenes including MYC and cyclin D1 will be activated. This event is rarely affected in the lung cancer. The dysregulation of WNT ligand transcription more frequently occur. For instance, the loss of WNT7A mRNA is frequently feature of some lung cancer cell lines and primary tumors. It is not only the WNT ligands, but also various signaling molecules that are dysregulated in the lung cancers. For example, overexpression of DVL3, a signal transducer molecule and positive regulator of WNT signaling pathways, was reported in 75% of primary non-small cell lung cancer. Then, the PCP signaling pathway can be activated by WNT11, which leads to the activation of the small GTPase RHOA, which is RAS homolog gene family member A, and RAC1. This will activate the JNK and ROCK that initiates remodeling of the cytoskeleton, thus leading to changes in the cell adhesion and motility. In small cell lung cancer, the RAS is overexpressed, which lead to the adenocarcinoma, while P53 and RB1 are inactivated. Next, the best known activator of calcium 2 plus or dependent WNT signaling is WNT5A. The activation of this pathway leads to a transient increase in cytoplasmic free calcium 2 plus level that can activate the PKC, CAMK2 and the phosphatase calcineurin. The findings that I found also in the murine cancer model, which also has been observed in the lung cancer. Recent study found that there is a KRAS mutation occur in human lung cancer. The KRAS mutations are missense mutations, which introduce the amino acid substitution at one of the positions 12, 13, or 61. The combination of KRAS mutation and activation of WNT signaling will increase the carcinogenic potential and the tumor size.
Okay, next I will present on abnormal pathway of lung cancer in haddock signaling. Okay, usually the dysregulation occurs in cancer when the lung cancer has inappropriate activation of this pathway, which is on haddock pathway. The location such as brain, skin, gastrointestinal tract, and prostate cancer. Sonic haddock more crucial in embryogenesis and it is broadly expressed gene. Furthermore, it is highly conserved in development. However, it is inactive in adult lung epithelium but active in epithelial progenitor stem cells. And there are two types of activation. One are via conical, which is SHH bind to receptor. Second is via non-conical, which the activation of GLI proteins independently. Next, we go to the function of the hedgehog pathway in human body. This pathway usually involves in formation of lung, continuous renewal of tissue, and repair of adult tissue, and the stem cell maintenance. Now we go to the significance of SHH in lung. First, it is essential for the growth and differentiation of trachea and lung. Second, it maintains the notch signaling pathway during the development and repair of the lung epithelium. Third, it regulates the bronchial bud formation when SHH secreted by epithelial cell with paracrine effect in mesenchymal. Fourth, increase the collagen synthesis in fibroblasts in three types of lung, which is newborn lung, normal lung, and also chronic disease lung. What is fibroblast? Fibroblast is to produce structural framework, which is trauma, and will aid in wound healing. Okay, I will continue on mechanism of sonic haddock, which is we call as SHH. So what happened inside the lung cancer? There is an increase in paracrine. Okay, paracrine is one of cell signaling type, and then the HH signaling from the tumor to the surrounding stroma. So what happened to the tumor to stroma signaling? There is been two mechanisms here. The first one is off and the second is on. So this is what we call as canonical pathway activation. Just now my friend have explained about type of the lung cancer. The first one is canonical. So canonical is binding, ligand binding of the SHH to the receptor. So what it do, the first one regulate the proliferation of cancer stem cell and to increase tumor invasiveness. The second one is reactivation in adult lung in response to injury which is such as tobacco use cause lung carcinogenesis. So we look at the mechanism here for the off pathway.
when there is the absence of the SH ligand, the PTC inhibit the activity of SMO, then the repress downstream. So next, if there is the binding of SH ligand, it will inhibit the PTC from repressing SMO, thus activate the downstream process. So next, we go to the expression of HH protein in non-small cell lung cancer, NSCLC. So there are two reasons here. The first one is normal lung epithelial, where moderate to high expression of SMO and PTCH1 were found in normal lung epithelial. And the second one is inside the lung tumor, where the low expression of SMO and PTC1 protein, but there is the high of GL1 were found in the tumor. However, they are considered as the overexpressed compared to the other tumor.
Okay, so for the next treatment for the pathway targeted, which is in this case WNT signaling. Okay, so we have concluded there is four treatment for for WNT pathway. One of them is the microRNA. So microRNA are endogenous, which are small non-coding RNAs that are able to suppress the gene expression by inhibiting the translation or increase the degradation of the target gene mRNAs. So we can, by referring the pictures, we can see there is the function of this microRNA specific for MIR34A, which is a thermosuppressor mRNA, microRNA, which is for in other name is anti-oncogene lah. So there is three specific microRNA, which is first is inhibit the beta-catenin activity. So for all you that you don't know, beta-catenin also referring to WNT lah. So MIR29 is down regulates beta-catenin expression, and lastly MIR T376C suppress the cortical WNT signaling. Okay, so the second treatment for the WNT pathway is which is Clodo, is a membrane that are known as the anti-aging epigenetic modulator that bind to WNT molecule to prevent the binding of the receptor. Okay, so the Clodo we can see referring to the picture. Focus only the blue one. Neglect the next of the two. So the middle one is the blue one. So the Clodo, our expression of the Clodo reduce active beta-catenin, thus inhibit the WNT signaling pathway downstream and reduce the cancer cell proliferation. Okay, you can see the beta catenin cause the tumor growth. So the overexpression of the Clodo reduce the active of beta-catenin. So the next treatment is wantitumab. So this is by injection you can use. And wantitumab is an IgG type of monoclonal antibody, which will bind to the certain receptor, which in this case is FZD receptor 12578 in the WNT signaling pathway. So it will preventing the activation of the WNT signaling pathway. Okay, so the WNT we cannot bind to the freezer receptor because there is wantitumab, the presence of wantitumab. So that's the result in the inhibition of the cancer stem cells CSC activity subsequent to inhibition of cancer cell proliferation. So you can see because the WNT cannot bind to the freezer receptor, so there is no beta-catenin, and so there is no the growth of the tumor cell. So the last treatment for the WNT targeted pathway is bevacizumab, which is bevacizumab also is an IgG type of monoclonal antibody that acts against the vascular endothelial growth factor, VEGF. That is a pro-angiogenic cytokine. So it acts the same as the wantitumab, which it will inhibit the binding of the VEGF in this case from the receptor binding, so thereby preventing the growth of the maintenance of tumor blood vessel. So because the prevent the growth of the tumor blood vessel, so there is no the blood will not be supplied to the tumor, so they will cut the growth of the tumor. So that is the function of bevacizumab. So we can apply this by the injection treatment.
Next, I will explain about the hedgehog signaling inhibitor. First is cyclopamine, which is it has high affinity for SMO, and upon binding, it inhibits the signal of hedgehog pathway. Cyclopamine derived from the word cyclovia, and also it isolated from the corn lily, belongs to the family of steroidal alkaloids. Second is cyclopamine tetrate. This protein have the characteristic which is water soluble, and the activity in inhibiting hedgehog signaling is higher than cyclopamine because basically due to the poor solubility of cyclopamine, synthetic cyclopamine was created to overcome it. And also this protein have the effective in causing tumor shrinkage in 2 mouse model of BCC. And next is itraconazole, which is antifungal drug and also safe drug and less adverse effect on human. And also this protein also for prophylaxis in immunosuppressive disorder. In inhibition of angiogenesis, itraconazole inhibits vascular endothelial growth factor signaling. With the step first is inhibiting the expression and glycosylation of vascular endothelial growth factor receptor 2, and which downstream target of hedgehog pathway through GLI1, thus suppress angiogenesis in cancer cells. In inhibition of SNO, ITS or itraconazole could act on SMO directly. With decrease the amount of SMO on the primary cilium to suppress the hedgehog pathway. And the last one is GANT61, which is small molecule inhibitor of glioma-associated oncogene 1, with interfere with GLI translocation into nucleus and can prevent DNA binding. And also this GANT61 interferes with the cell cycle by induction of G1. Last but not least, these have decreased level of BCL2 or anti-apoptotic.
As a conclusion, the major pathways and molecular interaction implicates the development of NSCLC and also the SCLC. The application of the treatment targeting a specific pathway such as WNT and HEDOC can be used in combating lung cancer by interrupting the growth of cancer stem cells through the inhibition of components in signal cascades. Combination therapy should be implemented for the future research of the cancer treatment. And this is the list of the references that we use in this slide. Thank you.